Hello. Cellular respiration is the metabolic pathway organisms use to transfer energy from organic molecules into ATP. Organisms are divided into two categories based on how they obtain energy: autotrophs and heterotrophs. Autotrophs produce organic molecules from inorganic molecules. For example, plants can convert carbon dioxide and water into glucose. Heterotrophs obtain organic molecules from the organisms they consume. We are heterotrophs that break down the organic carbohydrates, proteins, and lipids from our food into monomers and then absorb those monomers into our bloodstream. Our cells then break down these monomers to release energy used to make ATP. Cellular respiration can be defined as the oxidation of organic molecules to extract energy from chemical bonds. All organisms use cellular respiration to make ATP. The ATP is then used to carry out endergonic reactions in the cell, to do mechanics like muscle contractions, or it is used in active transport mechanisms. Excess energy is stored in the bonds of carbohydrates or stored long-term in lipid molecules. The cellular respiration pathway is a complex set of oxidation and reduction reactions, nicknamed redox reactions. These reactions slowly break down organic molecules in a stepwise process. A key reaction in this pathway is the dehydrogenation reaction. Instead of just losing an electron, in this oxidation reaction, a whole hydrogen is removed. The hydrogen is separated into a hydrogen ion, which is just a proton, and an electron. The energy needed to make ATP comes from these high-energy electrons. Of course, we know that when you have an oxidation, you also must have a reduction. Something must hold the electron until it is needed. This is the role of the coenzymes NAD and FAD. This image illustrates how these coenzymes assist in the dehydrogenation reactions. This energy-rich molecule is one of the intermediates in the respiration pathway. This molecule attaches to the enzyme and hydrogen are removed. The coenzyme is there to accept the electrons that are released. In this step, the two electrons get accepted by NAD. NAD also accepts one hydrogen ion and is reduced to NADH. The second hydrogen ion is released. FAD does the same thing, only it can accept both hydrogens, so it becomes FADH2. These coenzymes essentially act as taxicabs or electron shuttles that move the electrons from one step to another in the pathway. In a later step, energy is harvested from these electrons to make ATP. Once all usable energy is removed from the electrons, they must be deposited somewhere. Different types of organisms use different molecules to accept the used-up electron. Animals use oxygen as the final electron acceptor, and this pathway is called aerobic respiration. Here is the summary equation for aerobic respiration. It's a balanced redox reaction. It shows you the initial substrates, glucose, and the required oxygen. After the glucose is completely oxidized for its energy, the cell ends up with carbon dioxide, water, and of course ATP. Just like all energy transfer reactions, some energy is lost as heat in this process. Before we go into the details of the steps of aerobic respiration, we need to talk about how ATP is made. There's two ways to make ATP. The first is called substrate level phosphorylation. It's a fancy word for something very simple. It simply means that during the pathway there is a phosphate that's removed from a substrate molecule in the pathway, and it's attached to ADP to make ATP. This process happens by the assistance of an enzyme. In this image, you can see the enzyme in the pathway takes one of the substrates, rips the phosphate off, and attaches it to ADP, making it ATP. This illustrates a step in this process that we're going to see shortly, where the organic substrate PEP becomes pyruvate or pyruvic acid. So phosphorylation simply means yanking a phosphate off one thing and sticking it on another, and it's done to a substrate in the pathway. The second way of making ATP is called oxidative phosphorylation with chemiosmosis. This is where ATP is made using a proton gradient and an enzyme called ATP synthase. I will describe this method later in the pathway. The aerobic pathway is often divided into 4 steps. The first one is glycolysis. Glyco refers to glucose, and lysis means to split or break. This step breaks glucose in half. The first set of steps of glycolysis are called the priming reactions. Glucose needs to be activated by 2 ATP before it can be split. This activation energy is the push to start this exergonic pathway. You learned about energy of activation previously. Each end of the glucose in its straight chain form receives a phosphate from an ATP. This destabilizes the chain and makes it easy for an enzyme to split the glucose in half into 2 molecules called G3P. During the energy harvesting steps, each G3P has 2 hydrogen molecules ripped off, 2 NADH are formed, and 4 phosphates are removed to form 4 ATP molecules by substrate-level phosphorylation. The G3P has now been transformed into 2 molecules of pyruvate. 2 NADH have been made, and there is a net yield of 2 ATP. Remember, it cost us 2 ATP to start the process. All organisms do this first step. It does not require oxygen or mitochondria. It happens in the cytoplasm of the cell. So all forms of cellular respiration start with glycolysis. Please watch the Part 2 video for the rest of the aerobic respiration steps, and thanks for watching.